Hi everyone, today we are going to talk about microbial genetic mutation. Mutation is a change in the base sequence of DNA which ultimately causes effects on protein synthesis. Before going into the types of mutation, let us have a look on the source of mutation. Mutation is caused by external and internal factors. External factors include chemicals and radiation, while internal factors involve error occurs during DNA replication where DNA fails to copy accurately. Genetic information for protein synthesis is stored in DNA. Then it is transcribed into mRNA, and from mRNA it is translated into protein. If one nucleotide is altered, this will affect the mRNA sequence and change the type of protein. The first type of mutation is point mutation. Point mutation occurs when a nucleotide base is replaced by another base in the DNA nucleotide sequence of a gene.
If one of the base adenine is replaced by guanine in DNA strand, in the complementary RNA strand, the base uracil will also be substituted by cysteine. This alters the amino acid from valine to alanine.
There are three types of point mutation: missense mutation, nonsense mutation, and silent mutation. Missense mutation is similar to point mutation, where there is a change in one DNA base pair that results in the substitution of one amino acid with another. There are two types of missense mutation: conservative and non-conservative. Conservative missense mutation happens when the new amino acid substituted is similar in function and shape to the amino acid being replaced.
For example, the change in nucleotide base causes changes of the amino acid serine to cysteine. They are of the same type of amino acid, which is polar, uncharged amino acids.
The second type of missense mutation is non-conservative. Non-conservative mutation involves completely different type of new amino acid added to the protein chain.
For example, the change in nucleotide base causes change of asparagine, which is a non-polar amino acid, to glutamic acid, which is an acidic amino acid. There are different types of amino acid, and this will cause severe effect on the final protein synthesis.
The second type of point mutation is silent mutation. This type of mutation does not affect the protein synthesis. This is due to the degenerate characteristics of codons, where one or more codons code for the same types of amino acid.
For example, if the codon changes from GUU to GUC, the final amino acid is still valine, as both codon codes for the same amino acid.
Next, nonsense mutation. Nonsense mutation is point mutation which causes RNA sequence to form stop codon.
For example, if one of the A is substituted with C, the complementary base pair in RNA will change to UAG, which is a stop codon. This results in shorter and incomplete protein translation.
Other than point mutation, there is frameshift mutation. There are two types of frameshift mutation: insertion and deletion. For insertion, an extra nucleotide base is inserted into the DNA base sequence, and this causes the whole base sequence after the insertion point to be shifted one place backward. As mutation occurs, one base is inserted into the DNA base sequence. This causes change in the mRNA too. As a result, the first codon codes for glutamine, the second codon codes for glycine instead of glutamine. The new polypeptide chain is different from original and lead to the production of non-functional protein. For deletion, one of the nucleotide base is deleted from the DNA base sequence of a gene. This causes the whole base sequence after the deletion point to be shifted one place forward. Let us show the effect of deletion. Cytosine is deleted from DNA strand. The base sequence after deletion point will shift forward. It also does same in mRNA. Normal amino acid code for glutamine is changed to arginine, hence it produces a non-functional protein.